An important part of prokaryotic DNA replication is the replication fork. Prokaryotes have double-stranded helix DNA structures. These strands run anti-parallel to one another. DNA molecules are made up of nucleotides composed of a phosphate group, a sugar group, and a nitrogen base. Nucleotides are held together by phosphodiester bonds. Prokaryotic DNA strands are circular, whereas eukaryotes have linear DNA. This has a direct relationship on the mechanisms that prokaryotic DNA can use to replicate, theta replication being the most commonly observed. To begin, initiator proteins attach at a critical point on the DNA strand, referred to as the origin of replication, or ORIC. This initiator protein helps to unwind a small section of DNA strand, forming a replication bubble with a replication fork on both sides. Next, DNA helicase binds to the lagging strand template, breaking weak hydrogen bonds that keep DNA strands together. Behind DNA helicase, single-strand binded proteins attach to the strands to stabilize them as the replication forks widen out the bubble. In theta replication, replication forks can move bidirectionally. As they move, tension forms ahead of the replication fork. DNA gyrase is a type of topoisomerase that helps relieve tension by cutting one or both strands of DNA, releasing tension, and then resealing the two relaxed strands. To elongate, we first need to add RNA primers. DNA polymerase can't interact with a bare strand. Therefore, RNA primers provide a 3' hydroxyl group that DNA polymerase needs to function. DNA polymerase III has high processivity, allowing it to add a lot of nucleotides at once. It adds new nucleotides to the 3' end, moving with the replication fork from the 3' to 5' direction, growing the new strand in the 5' to 3' direction. The strand where it can do this continuously is called the leading strand. However, it also has 3' to 5' exonuclease activity, meaning it can occasionally jump back to replace erroneously added nucleotides. Following DNA polymerase III is DNA polymerase I. It moves in the same direction as DNA polymerase III and has exonuclease activity as well, but low processivity. Therefore, its primary job is to remove RNA nucleotides and replace them with the correct nucleotides. Opposite to the leading strand, the lagging strand runs in the 5' to 3' direction. In the lagging strand, DNA polymerase III can only move 3' to 5', causing it to move in the opposite direction of the replication fork. For DNA polymerase to work here, several RNA primers are required to guide DNA polymerase. This results in fragments of replicated DNA strands being formed, referred to as Okazaki fragments. To join the Okazaki fragments, DNA ligase will swoop in and glue the fragments together by adding a phosphodiester bond between the nucleotides added by the DNA polymerase III and the nucleotide added by DNA polymerase I. As the replication forks travel around the circular DNA, it will eventually engage in termination where DNA replication is completed.